We have seen already how to assess if a gene is differentially expressed using t-test and the concept of p-values. So just to remind you what we did here, if we had two classes, for example infected and control, and we have some measurements in each class. So this is the expression level here, and some other measurements here. Then we can calculate the means and the variances of this sample and this sample, and variances. Calculate the t-statistic then, and then assess whether this t-statistic is significantly different than the value 0. Because under the null hypothesis, μ1 equals μ2, which means that the t-statistic is 0. This is the null. Okay. When we analyze RNA-seq data, then we usually end with counts of reads for each gene. Now, it is important to understand that when we deal with count of reads, these are integers, we cannot really apply the t-test. Another reason is that for several genes, the number of counts, or the number of reads, is very, very small. And in this case, the variances that has to be computed, for example in t-statistic and in statistics in general, cannot be computed accurately. So the estimation of variance when the number of reads is small is not accurate enough. Therefore, we don't really apply the t-statistic and the t-test on RNA-seq results. Another reason is the following. Okay, let's see now how we will able to process immediately the number of reads. So assume that we have done an RNA-seq experiment. And we have obtained in total n reads. So these n reads are actually reads that are mapped to different genes. Like this, for example. So this is gene 1, this might be gene 2, and so on. So in total we have n reads. Now in this cell that we are studying, or in this organism, let's assume that the expression level of gene 1 can be measured with this number p, which p here is the probability that this gene is expressed. Or in other words, from the total RNA, from all RNA, mRNA in the cell, what is the proportion, or what is the frequency of the mRNA from gene 1. So what I want to say here is that this P will be this quantity here, mRNA from all cell, from all genes, and here it will be the mRNA from gene 1. So this is actually the gene expression level of gene 1 compared to the total expression level. And it is actually a parameter, because this is the real expression level of gene 1, and this quantity is actually unknown. Now, if we do an RNA-seq experiment, what we actually do, as I have shown here, is some number of reads from gene 1 and some number of reads from all genes. Now, for this specific organism, the number of genes for each, the number of reads from gene 1, so this quantity here, let's use the symbol G1 reads. So the G1 reads will actually be a random variable, and its value will depend on this P. You can understand it in the following way, that this is the actual expression level of gene 1. Now you can make a library of n reads, you can measure the reads in G1, and you might find the number, let's say 1000 reads in gene 1. Then if you repeat this experiment exactly with the same parameter, the same expression level from gene 1, you might find 1020 reads for gene 1. If you repeat the experiment, you might find 980 reads for gene 1. If you repeat the experiment, you might find 1100 reads for gene 1, and so on. So repeating these experiments, even though the expression level of gene 1 is the same, the real expression level is P, you don't find the same reads from between replications, just because the number of reads is a random variable. Okay, but the good news here is that this random variable follows a specific distribution, and follows a Poisson distribution with parameter P multiplied by n, where P is this parameter here, is the expression, the relative expression level of gene 1, relative compared to the whole genome, to all genes, and this n is the number of reads that we have taken from the whole genome, from all genome. And realizations of this process might be these numbers here that I have written. Now, this Poisson is a Poisson process here, as a Poisson distribution here. It says that what we expect to see is P multiplied by N reads. This is our expectation for the number of reads for gene 1, and the variance will be again the same. So this is a realization of this Poisson distribution. You see that the mean and the mode here is in 1000, and there is some variance, and the variance here is exactly the same as the mean. Okay. Now, this variance describes very well the variance from such a process if the P, this parameter here, between replications remains the same. So as I said in the beginning, if we do this experiment one time and then we repeat it another time and then a third time with the same parameter P, then this is a Poisson variable. But when we have real samples, what we actually do is that we measure the expression levels, so the number of reads here, between individuals, different individuals. So for example, in the experiment that we discussed in the class, that we have infected C. elegans, we have 3 different C. elegans animals. 1, 2, 3. And for each one of these 3 animals, we measure the number of reads for the same gene. Now, besides the and we observe different number of reads here. So for the gene 1, let's say that we observe G1 one number of reads here, and G2 one number of reads, and G3 one number of reads. So this index 1 to 3 refers to the animal. Okay, and a part of the difference between these measurements is because we repeat the experiment 3 times. So we see here that we get different realizations of this Poisson process. One time we get 1000, the second time 1020, the third time 980, and the fourth time 1100. So a part of the difference is because we repeat the experiment 3 times here. I will just write here replications of an experiment, and because the number of reads is a random variable, then we will get a different result every time of an experiment. And number 2 is because there is a biological difference between the individuals. Which means that we cannot say that there is a constant P for all these 3 guys here, which means that there is a P1 for the first individual, a P2 for the second individual, and a P3 for the third individual. So these 3 repetitions is not actually repetitions of they cannot be described with a Poisson distribution, because this probability of expression for gene 1 is not the same for all these three individuals, and it's not the same because there is some biological difference between them. They are just not the same individual. Okay, therefore what we can do to model better this difference of reads between these three individuals is to assume that the p between individuals is not constant, but follows some distribution. So what they have done, and I will not go so much into the math here, what they have done is that they have assumed that this p follows a gamma distribution with parameters alpha and beta. This is not really important. And if this is the case, so if this p follows a gamma distribution between individuals, then these numbers, the number of reads between individuals now, follow a negative binomial with parameters alpha and beta here. Or to put it in other words, the number of reads, which will be easier I think to understand it, follows something like a Poisson, but this probability, this is the rate of the Poisson, this is lambda equals this number, but this number here is not constant, but this number follows a gamma. It's just not constant. You can assume that this is not constant but follows a distribution that may look something like that, let's say. So this number with parameters alpha and beta here. And with this way, the mean is p by n. Let's say that this equals to lambda, or yeah, let's say that this equals to lambda, but the variance now is lambdas plus lambda square phi, which is a positive number. So we see that remember that the variance of the Poisson was lambda, so compared to the Poisson, this process has a greatest variance, and this greatest variance is split in two parts. One is like the Poisson, and it's the technical replicate, so if we could just repeat the experiment 3 times with the same from the same individual, we would get a different number here, and this is attributed to this lambda. But there is a second part, which is this one, and this actually reflects the biological difference between the individuals. So this is what EdgeR and similar software like Dissect2 are trying to achieve, and that's why this software is better used for the differential expression analysis of RNA-seq measurements. Now if we go to Galaxy, where we have done our mapping and we have used htSEQ to count things, here I have used the function column join in order to create this table of results, where on the rows I have the genes, the gene names, and on the columns I have the 6 individuals from which I got measurements, I got expression values. So this number here, for example, represents the number of reads that I got from the first individual for this gene. And what we must mention here is that the first 3 individuals are actually infected, C. elegans, while the 3 last individuals, the 3 last columns, are individuals that are controlled. So I'm interested in detecting genes that have differential, the genes that behave differently in the first 3 columns compared to the last 3 columns. Okay. So when I use a jar to analyze this matrix, let's go here to see how to do it. So as input, I use this column join on the data that I have. There is no information in the file regarding the factor that I'm testing. So what I'm testing here is to see if the infection has any role on the expression level, and within the file there is no such information regarding infection or non-infection. There is no information of on what I'm studying. So this means that I have to put everything by hand. So here in the subsection factor, I must write what I study, and what I study is the role of infection on the expression level. You can assume that this factor here is a discrete random variable that may take some discrete number of values, and the name of this random variable is infection. And the biological meaning here, or the meaning of this variable, is what actually I am studying, and I'm going to study the role of infection on the expression level. And this infection, this factor here, may take 2 values in my experiment. The first value is the inf, meaning infection or infected individual, and the second value is control, meaning non-infected individual. So in this experiment I have just one factor, but I may also have more factors that I can insert using this button here. Okay, and the way that I must put the information for the levels is that I must assign a value to each column. And since in my experiment I had 6 animals, I must provide 6 values here. So the first animal is infected, 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 and the 4th animal, 5th and 6th are controlled. Okay, I don't have gene annotations, and regarding these levels here, what I want to test is if there is a difference between infection and control. So in other words, if the mean expression level of infected minus control is different than 0, and that's why I put it in this way, inf minus control. So the order here does not really matter, but it matters later on the interpretation of the results. Because if I find that a gene is overrepresented, it will mean that it has higher expression values in the infected animals compared to the control. If I would have put this in a different order, overexpression would mean that it has higher expression values on the control compared to the infected. Okay, so everything is here. I can also put some filterings if you want. Filter low expressed genes. Usually I must put a yes here, and it has some additional values. CPM, minimum CPM. So CPM means counts per million, and this means that if so many samples, so 2 samples have lower than 1 CPM, so I have 6 samples. If 2 samples out of the 6. If at least 2 samples out of the 6 have an expression have 1 count per million for the particular gene, at least 2 samples out of 6 have at least 1 count per million for the particular gene, then I will keep it. If only 1 sample fulfills this criterion out of 6, or if no sample fulfills this criterion for the specific gene, then I will not analyze a specific gene and I will reject it from the analysis. Usually it's a good thing to filter out very lowly expressed genes. Okay, and now I run the tool. So the analysis has finished now, has been completed, and we are ready to see the report. I will click here on the report tab of EdgeR. So EdgeR outputs 2 files, one is a report and the other one are the tables. We will see the tables later on. So regarding the report, let's see first the biological coefficient of variation, which means how much variation EdgeR detects within each of the 2 groups as a function of the expression level of the genes. So this means that within each group, so within the infected and within the control, I stress the words within, genes that are lowly expressed have higher biological variation. They are more variable between the individuals. So between the 3 individuals in a group, there is greater variation for genes that are lowly expressed. And this is something that we know for RNA-seq experiments that it's true. So genes, the variation is greater for the genes that are lowly expressed, and that's why we don't really trust these genes, the measurements of lowly expressed genes, so much. And then this increases, or the variance decreases as we go to higher expressed genes here. This is another measurement of similar things, and the other important plot here is this plot that shows the log-fold change as a function of the average expression. So log-fold change here is the following measurement. I have the 3 individuals, individual 1 to 3, that are infected, and I have the 3 individuals from the control. From each one of them I have an expression measurement, let's say 30 reads, 20 reads, 30 reads, and here I have 10 reads, 5 reads, and 6 reads. So the average value here is so the average value here is about 26 and here is about 7. So the log-fold change will be a logarithm of 26 divided by 7. And since this is greater than 1, this value will be positive and maybe quite positive. Okay, and the log of C is useful because if it is negative, means that in this class we have under-expression compared to this class, and if it is positive, it means that in this class we have overexpression compared to this class, as we have here. Okay, let's go back to the plot now. So these are, all the black dots here are the log FC for all genes. And you see that there is a pattern here that for lowly expressed genes the log of c might be very large or very small, and then as we proceed we have smaller deviations from the 0. I must say here that if this is 0, it means that log x equals 0, it means that x is 1, which means that the average expression value here is the same with the average expression value here. So log infected to control is 0, which means that infected to control equals 1, which means that infected equals control for the value 0. Okay. However, since we don't trust really so much the genes that have small expression levels, we must be more strict when we consider that the gene is differentially expressed if it is lowly expressed. So for genes that have low expression levels, like on this side, so here for example, we must observe either a very positive log of c or very negative log of c in order to assume that is differentially expressed. And if we go towards higher expression levels, then we can assume that we have differential expression even for smaller deviations from the 0. For example, this gene here is differentially expressed even though it is not so much higher for the infection class compared to the control class. You see, that's not very far from 0. While this gene here is differentially expressed, but it's the average expression level in the infected class is huge compared to the expression level in the control class. Okay. Now another result here is the PCA, which shows how my individuals, the 6 individuals, can be represented in the in a plot here using the principal component analysis. In fact, here is not a principal component analysis, is the multidimensional scaling plot. It's similar. It shows we try to represent how these individuals differ from each other if we would take into account all the genes. And we see that the first 3 individuals, so let's see, the labelings to 2, 3 and 5 are 2, 3 and 5 somewhere here. 2, 3 and 5 are infected and the rest are control. So these ones are very, very different than this one, which means that several genes have been affected in the infection class compared to the control class. Now this plot is useful because if we have several samples, it might show that in one of these classes we might have another subgrouping. For example, it could be that we have several points here and several points here, and this could mean that maybe there is another factor that we have not taken into account. If this is happening, then usually what we observe here in the BCV plot, the biological variance, biological coefficient of variance, is that here the variance, this coefficient of variation will start to increase again in the end. So using this plot as diagnostic tools, we must be careful to not observe a huge grouping within each of these classes, so that we have 2 groups of control guys and 2 groups of infected, or 3 groups or whatever. And here we should not observe an increase for genes that are highly expressed. If this is okay, then we can trust actually these results. If not, then we should take into account an additional factor that we haven't taken yet. Okay, and this, if this is fine as it is here, we can now go to see the results. So I go here to the table, I click here, and what we observe is the following table. So here everything is ordered according to the p-value. Genes that are more differentially expressed are first, and then we have genes that are not so much differentially expressed. So the first column is the gene name, the second column is the log-fold change. For example, this gene here means that it's much higher expressed in the infection class compared to control class, while this gene here, it means that it's higher expressed in the control class compared to the infection class. This log CPM is the logarithm of the counts per million. It shows how much the gene is expressed on average, and here we see that this gene is quite highly expressed. This is the statistic that they use to assess differential expression. As with the t-test we use the t-statistics, so here they use another statistic, the f-statistic. This is the p-value for this statistic to be different than 1, I think. So this is the p-value that refers to the null hypothesis that the infection class is the same as the control class. And because we compare many genes, they do a process that is a correction for this multiple testing. We're going to find the differential expression for many genes, and this results in a corrected p-value, and this is the last column here. So usually when we want to assess which genes are differentially expressed, we go to the last column and we get all genes that have a lower p-value than a certain threshold, usually the 0.05. So all these genes here are differentially expressed. Many actually, based on these measurements here. So several hundreds of genes are differentially expressed. Somewhere here should be the threshold. Yeah, 0.0, yeah, somewhere here. Okay. Now these genes are split in overexpressed genes and underexpressed genes, like this one and this one, and the next part of the analysis is separately to examine the higher expressed genes and the overexpressed genes and the underexpressed genes, and we'll show that in the next video.